Mathura. I extend a warm welcome to all esteemed dignitaries, respected speakers, faculty members, researchers, and participants joining us today for the 6-day online faculty development program on exploring innovation in pharmaceutical sciences for better future. We are delighted to have such a distinguished gathering as we begin day 2 of this academic journey. So, without further delay, first of all, I would like to I take the privilege to invite Dr. Yogesh Murthy, Associate Professor, GLA University, Mathura, for address the gathering. And first of all, I would like to read the profile of Dr. Murthy. Dr. Yogesh Murthy is an associate professor at the Department of Pharmaceutical Chemistry, Institute of Pharmaceutical Research, GLA University, Mathura, with 18 years of teaching experience. He has
of Pavanji.
impressive academic record, having published in renowned national and international journals, along with 6 books and 14 book chapters, and editing 1 book. Dr. Murthy holds 2 granted Indian patents and 6 published patents, and has successfully supervised 12 MPharm students and 1 PhD scholar. He is a life member of several prestigious professional organizations, including APTI, IPGA, ISTE, ISCA, ACPI, CSI, IRCS, and Farm Association. Dr. Murthy is member of IPES, Society for Ethnopharmacology, Vigyan Bharati, and serves as a nominee of IAEC and by CPCSA. Dr. Murthy excellence in teaching and research has earned him awards such as Most Promising Teacher in Pharma Education 2019, Star Performer 2020, Outstanding Oration Award 2021, Outstanding Teacher Award 2022, and recognition for extraordinary performance during 2022, as well as most popular contained by the UP Higher Education Service Commission in 2022. His main areas of expertise include phytochemistry and synthetic chemistry. And I request, without delay, to our co-convener of this faculty development program, Dr. Murthy, please address your speech. Sir, kindly address the gathering, please.
Thank you, Pavan. Thank you for giving a nice introduction. Ladies and gentlemen, esteemed faculty, and dear participants, I, Dr. Yogesh Murthy, co-convener of this e-FDP, feel pleasure to welcome you all to this online faculty development program on the theme Exploring Innovation in Pharmaceutical Sciences for Better Future, organized by IPGA Mathura Local Chapter in association with Rajiv Academy for Pharmacy, one of the oldest institute of Mathura region. I feel proud that Rajiv Academy for Pharmacy, my alma mater, took the challenge to organize this e-FDP in a very short notice. Now come to the theme. In today's rapidly evolving world, innovation is the key to unlocking new possibilities and addressing the complex challenges we face in healthcare. This FDP aims to bring together experts, researchers, and educators to share their knowledge and insights on the latest innovation in pharmaceutical sciences. Over the week, you will explore cutting-edge topics, discuss emerging trends, and exchange ideas on how to harness innovation for a better future in healthcare. I would like to express my gratitude to our esteemed speakers organizers and participants for their contribution to this FDP. And I am confident that this program will provide a valuable platform for learning, networking, and growth. I wish you all a fruitful and enriching experience. Radhe Radhe.
Many thank you, sir. Thank you so much for your insights and valuable motivating words. So, without any delay,
Your mic working, I think. We
I
cannot hear you.
Pavan, unmute yourself.
our eminent speaker,
Sunasa, your voice is audible. Don't worry. But Pavan's voice is not coming.
sir.
You can continue, Pavan. Continue.
Am I audible now? Na?
Yes.
Okay.
Yes, Pavan, you are audible, yeah. Yes.
Okay, sir. Thank you. So, if I am talking about the brief profile of Dr. Suman Sinha, sir, Dr. Suman Sinha is today our eminent speaker. Dr. Suman Sinha is an assistant professor at GLA University, Mathura, with a focus on computational biophysics. He uses computer simulations and machine learning to study how protein interact with other molecules, which helps in drug discovery and understanding biological system. He completed his PhD in pharmaceutical technology in 2017 and has more than 10 years of experience in teaching and research, including a postdoctoral position at TIFR Hyderabad. Dr. Sinha has published many research papers and works with top research institute. He is also a member of the editorial board for Cancer Reports and reviews articles for scientific reports. His works strongly contributes to the progress of pharmaceutical sciences and drug development. I request to our eminent speaker kindly address your words. Sir, please enlighten us. We are privileged to have Dr. Suman Sinha, sir.
Start. I am sorry, I cannot hear Pavanji's voice. So he's not audible to me.
But you are audible, sir. Please continue.
Shall I start the presentation?
Sure.
Yes, sir.
Sure.
Yes, sir. You are audible, sir. You are completely audible.
Stop your presentation and unmute.
Good evening, dear colleagues. Thank you so much for inviting me to deliver a talk at this prestigious avenue. I take this pleasure to share some information which I have gathered over the years, and it's about my understanding of the subject. So, drug discovery, as we all understand, is a completely interdisciplinary domain where computational science, physics, chemistry, biology, all these areas of science, they share their own contribution in this field. So as I in my
मैंने
career,
स्कूल में नहीं करा, मैंने यहाँ कॉलेज स्कूल में तो जो मेरा बायो वाला 12th था ना वो तो ऐसे ही था। दीदी जा रही चाय छान के ला रही ना, नहीं बहुत तो है ही नहीं, पहले तो हटा दी, चाय बन गई यहाँ वैसे ले आ रही है।
Yeah. Okay. So I started in my PhD. I used to do three things: computational, and then organic chemistry, and then at the end I did some.
Request to all, kindly mute your mic, please.
Over the time, throughout my postdoc days, my interest grew more towards computational techniques. And as a result of that, I have completely left, I would say, experimental approaches like organic chemistry and biology, and I decided to focus completely on computational techniques. So that's what my research is. Even if I collaborate on using these approaches. Having a background of experimental techniques, it gives me kind of leverage to understand what my collaborators are talking about, understand their results, and then collaboratively we try to publish a paper, if at all a paper. So from bites to breakthroughs, computational drug discovery in the AI area. Something very interesting is going on around the world where things are getting changed at a very fast pace, let alone drug discovery. Everything is changing in a very fast pace. And of late, artificial intelligence and machine learning has impacted us in substantial ways. And if we talk about drug discovery is not far away. We see trends like traditional drug discovery companies. They are collaborating with AI startups to throw some insight into their early stage drug discovery campaigns. Even I created this image, which you see in the first slide, using a AI platform called Nano Banana from Google, the latest AI model from Google. So as you can see, it has impacted us in every sphere of life. Without any further delay, let me share what I understand from what's going on around. So I'll be talking about like traditional drug discovery, the challenges of traditional drug discovery, then how the computational methods have evolved over time, then what the AI is, how the AI is impacting, and the key technologies and breakthroughs. And then, of course, I'll talk about some success stories and case studies. And like every other technique, AI does not give a solution to every problem in drug discovery. It has its own challenges. So I'll talk about it. And of course, in the final thing, I will talk about the future directions and what are the opportunities it creates for all of us. So what is the traditional drug discovery challenge? It's drug discovery is basically two types of approaches traditionally we follow. One is target-based drug discovery, where we know that a drug is binding to which protein. What are its targets? It can be one protein. It can be a protein-protein complex, or it can be a DNA. And similarly, phenotype-based drug discovery, like ligand-based drug discovery, also it is called, where the traditional approaches like USAR, like Hans model and all these things, although we have come a long way after that. And then pharmacophore development, pharmacophore models. So typically we start with 5 to 10,000 compounds where we have done some initial screening, and then we have come to 5,000 compounds, which takes about around 4.5 years to do this initial screening. And then after within that, there is like things associated like biological assays, everything, synthesis of those compounds, biological assays. So around it takes 4.5 years. And then we move to the preclinical stage where we do pharmacokinetics and pharmacodynamic studies before it is being tested in humans, which deals with around 250 to 300 compounds and takes around a year. And then comes clinical trials, the most rigorous part of the drug discovery. Then we have in the clinical trials, we have phase 1 to 4, where 4 is post marketing surveillance. In phase 3 itself, it is marketed. After clearing phase 3, it is marketing, and phase 4 is post marketing surveillance. So phase 1 is safety in humans, they check, and then the effectiveness at treating the disease. So safety is most important. So toxicology comes into picture. And at this stage, phase 1 to phase 3, we typically deal with 5 compounds, 5 to 10 compounds, and we pass those compounds through this clinical trial screen, and which takes around 6 to 7 years. And then finally, we get one approval, which takes around 1.5 years. So if we add these years, 4.5, then 1, then 6.5, it takes around 12 to 13 years. It's a very long time. In 12 to 13 years, technologies change. We have seen that. So this is the major challenge, time. Another challenge is toxicity. So all these things, these are the pertinent problems. These problems are still there. We have not solved all of these problems. And then 10 to 15 years, money is around $3 billion US dollars in an average, including failures. We spend. The success rate is, success to failure is 10:90. 10% of the molecules can hardly pass through the clinical trials. Then, of course, high regulatory and safety hurdles, it is there. And then the limited chemical space exploration. In chemical space exploration means, chemical space means what? Chemical space means the number of, not the number, the chemistry known to us. The synthetic routes has been already established. So that is the chemical space. That is
Please unmute yourself. Please, ladies. Please, ladies, unmute yourself. Yes, sir, continue.
Yeah, thank you so much for intervening. So 90% failure rates, around $2 billion already spent, and the time it takes 10 to 15 years. So that makes drug discovery a very risky proposition, even for big pharma companies. No wonder we don't see it in India. In India, hardly drugs are discovered, if not, no drug has been discovered in India. So it's due to this money, the time we spend, and there is no guarantee that you will get a success. So evolution of computational method, when computational methods, initially how it was done is people used to synthesize randomly And then for all those chemical moieties, they used to check on animal models, and it leads to a lot of lethality of those animals. And people didn't have the idea of, targets where this molecule is going to bind, how this molecule is going to act. We were clueless. People were clueless till 70s and till late 70s and even in 80s. So in 80s, around a technology called X-ray crystallography of proteins, biomacromolecular crystallography. It came into picture, and then with that technology, we came to know that yes, proteins can be crystallized, and there lies inside the proteins, there lies a very deep pocket called active site where the drug usually binds. So the whole world was moving towards a structure-based drug design. It's called structure-based drug design. So how it typically how they used to do, and still it is done, and in the beginning stages. So molecular docking and virtual screening we do. Then suppose we get very good compounds, and then we pass it through X-ray crystallography to make a co-complex with the proteins and NMR. And then there was a ligand-based drug design approaches where QSR modeling and pharmacophore modeling was there, and then similarity-based approaches where this ligand-based drug design basically deals with history. A lot of compounds has been synthesized, and their biological activities has been already reported. So we make a database of all these compounds and biological activities in terms of IC50 or KD or KI, whatever metric. So and based on that, we used to do regression models to find out what is the common features. So what makes a good drug? What makes a bad drug? Based on those approaches are still there. Those approaches are still there, and those approaches are still used even in AI. So they are very powerful approaches. And then high throughput screening. High throughput screening means I take a lot of compounds and then I pass through a protein target experimentally. I'm saying protein target, and then we based on that we decide compounds. So docking and then similarity search, and nowadays ML-based approaches are coming into picture. I will talk about it after some time. And so basically when you have a ligand and then you have a receptor, and then you tell the software to dock the ligand inside the active site pocket, and then based on the docking scores, which essentially gives us a kind of hint towards the Gibbs free energy of binding. From there, we select the best pose, and then we select the best compound. And taking this complex, then we run MD simulations, which kind of talks about the stability of that particular drug molecule, that particular chemical entity inside the active site pocket of the protein, how stable the compound binds. Needless to say, a good compound will bind for a longer time, and a bad compound will come out in a very less time. And then we had QSR, where, for example, there is a database called Campbell, where the structures and the biological activities are reported. It's a database, so it's a public database. Anybody can take data from Campbell, and then they can do a data cleaning and filtering, and then they calculate the descriptors. Descriptors means the properties of the molecule. Molecular weight can be one descriptor. Number of chlorine atoms in the molecule can be one descriptor. One molecule has one chlorine atom, another molecule has 3 chlorine atoms, so it becomes a descriptor. And then based on that, we select features, and then we split the data set, and then we try to construct a QSR model, and then we see whether the model is able to reproduce the biological activity, not in real terms, but the trends of the biological activity. Then we validate the model, and then based on that, we screen more molecules. Similar pattern of QSAR has been applied to pharmacophore, where the number of descriptors in QSAR, where it was thousands in thousands, in pharmacophore it is only 1, 2, 3, 4, 5, and 6. Only 6 features, for example, hydrogen bond acceptor, hydrogen bond donor, anionic, cationic, and hydrophobic functional groups and aromatic functional. And what is a pharmacophore? Pharmacophore is basically a 3-dimensional arrangement of all these features that fits to all the molecules, or how many molecules it fits. So it depends on that resultant statistics, which gives us an idea of the quality of the pharmacophore model. So basically an arrangement. So this is how computation drug discovery has been going on since till last 3-4 years. And slowly now it is making a paradigm shift, although the basic calculations are still being done. So what are the limitations of traditional approaches? See, what I understand, the softwares are completely based on physics-based principles. And in physics, which is very straightforward, but in biophysics, where we have to deal with a lot of water, a lot of ions in the cellular environment, pH differences are there. So what happens is the miss is still very high. We cannot guarantee that any molecule we screen in using a computer that actually will bind very strongly to the protein in experimental validation. There is no guarantee. And this is one of the limitations. And then very limited exploration of the chemical space and the poor prediction of the drug behavior in vivo. Traditional approaches, computational approaches, we cannot say how the drug is going to behave when inside the human body or inside the animal body. So, and again, difficulty in handling complex biological systems. Suppose there is a protein complex of 2 proteins or 3 proteins even. So it becomes difficult for the computational for the docking program to find out to get a proper result. I myself have seen many failure cases, although I have seen very good success rates, but the failures were also throughout my experience in my career. I have seen a lot things fail. And then high computational requirements for complex simulations. I'll just give you an example. One good computer to run MD simulation costs more than 5 lakhs. So how many people in this country, they have money to buy those computers? And in very good pharma companies, they have thousands of these computers. So the one who is having resources is going to get the success because he will run it many, many times. And then you want to be sure whether you're going to get it or not. So in the perspective, I suggest you to remember the remember the amount, 2 billion US dollars, which is a huge money. So they can spend 1 million for computational resources. But for us, for academics, it becomes very difficult. So, but we have seen cases where academic research faculty, university faculty, they have come up with drugs. They have filed patents. Their molecules are going to, clinical trials. We have seen that. Example, I remember there is a professor in University of Southern Australia. His name is Von Easteen. So Von Easteen, he discovered 2 drugs, oseltamivir and zanamivir. So it is antiviral drugs for H1N1. So success is there, but as we see, a lot of limitations are still there. But nevertheless, it gives us a kind of leverage. Rather than doing things blindly, let us calculate and do things. So we are guessing but we are doing we are making a calculated guess. So that calculation is done by the computers. So the AI revolution, which started around the revolution, if we say, the science was there, the techniques were there, but the revolution started 2, 3, 4 years back when people wanted to do more robust calculations and find out features, find out trends, that this is a good trend, that this is a bad trend, and what is the trend we should follow, all these things. So key AI technologies transforming drug discovery is these algorithms are very famous nowadays. For example, neural networks and pattern recognition, and then natural language processing for literature mining and knowledge extraction. Then, of course, for molecular representation and interaction modeling, we have graph neural networks. And then generative AI and reinforcement learning we use for de novo drug design and optimization. So we can actually tell the AI to design a molecule for me. Design a molecule for me, de novo, novel molecules. The molecules which have never been, synthesized in the lab before. So it uses contemporary knowledge of chemistry, and they are so intelligent now that these programs can, like generative AI, they can actually generate molecules. So AI application across drug discovery pipeline. For example, in the drug discovery pipeline, it starts with a target identification, that which protein I am targeting, exactly what that protein does. We can use multi-omics data analysis and biomarker discovery in using AI. And then we can use lead discovery and optimization using virtual screening of billions of compounds. There is a technique called diffdoc. You can try diffdoc. It's free to use, and it's very nice, and it's very fast. The major advantage which AI gives us, it is the speed and the accuracy. In virtual screening of billions of molecules, and then we can develop QSR models using AI algorithms like random forest or deep boost, or for all boosted models, neural networks. We can develop QSR models, and then of course ADMET prediction. We can predict toxicity, and we can use the drug behavior using AI models. Clinical trial design also, they are saying, I don't have much idea, that they use AI to stratify patient and outcome prediction, all these things. These things are all based on data. So AI works on data. AI works on statistics. If you have data, then the AI will be able to predict things more efficiently. Statistics does, basically. But AI does it in a more efficient way. I won't say error-free, but less errors. Less errors. So yeah, these are the key breakthroughs and technologies. What do we have? Most important contribution of AI in recent times is most probably AlphaFold. I hope everyone has heard about AlphaFold. AlphaFold has come from Google's company called DeepMind, and where they have generated this algorithm called AlphaFold, which can give us a structure of a protein if we give the program, if we provide the sequence, the amino acid sequence to the program. It does not exactly says how the protein, what is the mechanism of folding, but of course it says what is the structure of the final protein. And that is, I would say, brilliant. 50% or more accurate than traditional physics-based methods. I have been working on a problem on a protein called SIG-E, which the structure of the protein, as reported in the protein data bank, it was in complex with a DNA. But when we are targeting the drug in vivo, it is not in complex with the DNA. So the structure completely changed. So I have used AlphaFold to predict the structure of the protein, and then we did some pocket prediction, and based on that we actually found out a few leads, and we are writing the paper now. Hopefully it comes up. So I myself have used. So I know how efficient it is. Rather than doing it blindly, let us make some, calculated guess. So it accelerates actually target identification and validation. And latest is AlphaFold 3. You can use it from, let me show you. You can use it from this platform called alphafoldserver.com. It is basically open source model, and people can accept it, access it.
Pavanji,
Yes, sir.
see what is the problem?
And there might be a network issue.
Oh, sir, there is a network issue, I think.
Okay.
I will contact to technical head. May they will help you. Sir, you are visible. Sir, am I audible?
Yes, Pavan, you are audible.
Yes, you are in now.
Can you hear me?
Yes, sir. Yes, sir.
Yeah, so I'll continue. So I don't know, some technical glitch happened. I accessed AlphaFold 3 with a different Gmail account, so that's how it got botched up. Sorry for that.
No problem.
So generative AI is creating novel molecules. It can create small molecules. It can create completely new proteins, and the humanity has never seen structures like this. And so nowadays, what is the thing we are seeing, that 80 to 90% of the phase 1 success rates versus 40 to 65%, 40 to 60%, let us say, traditional approaches.
Your presentation is not showing. Share your presentation, please, again.
Yeah. Okay. Enter screen. Wow. Can you see it?
Yes, sir. Now it is visible.
Okay, thank you. Yeah, creating novel molecules is again is a forte of generative AI, where we see the phase one success rates are 80 to 90%, which was 40 to 60% in traditional approaches. So we get a 30 to 40% boost in this success rates. So what machine learning methodology is, how it works, like in lead optimization, for example, in generative AI, not in generative AI as such, in random forest or all these models, how they're working. So like in traditional approaches, we collect the data and then we do a feature engineering cleaning, we normalize the data, and then we choose the algorithm. Then the algorithm gives us a model. Then based on the statistical parameters it provides, we assess the model, and then we go for descriptive calculation and model assessment, and then lead optimization. And from lead optimization, final admit prediction we do. This in lead optimization to admit prediction, also AI models are applied. For example, BIS, PK, KD, PK, PD models are there. Toxicity models are there. From there, solubility models are there. So we try to, based on past data, we try to, understand. And then finally we do a docking simulation, and we go straight away go for clinical trials. Okay, another thing is molecular dynamics. It has been a big problem. As I was saying, like a computer which costs 5 lakhs, how many of us can afford to buy a computer in our academic institutions? Not many of us. For this, we need huge funding from government, which currently is not there. So there is a technique which came up from Microsoft. The technique is called BioEMU. Again, it is as simple as you give a protein sequence to BioEMU, and then it creates, as you can see, it creates different structures of that protein just the way molecular dynamics does. So it helps us in understand molecular mechanism. It helps in structure-based drug design, and then it predicts properties like what will be the binding energy of the particular molecule with this protein target. The error rates are within the range of 3.5 kcal per mole. So we can say it is negligible compared to traditional approaches. So BioEMU actually, it is actually a very big AI model, which is based on 200 milliseconds of molecular dynamic simulation data, and then 500,000 experimental protein stability data, and then a structure of molecules predicted from AlphaFold. So finally, we get something called BioEMU. A kind of a protein of this size, if we start from the sequence and fold it like this, it will take us 10 GPU years, almost 10 years. So 10 GPU years, and the free energy surface of the molecule it looks something like this. So free energy surfaces are nothing but these are the energy minimas. The blue regions are the energy minimas, and the red and yellow regions are the maximas. The energy, free energy, is very high. So, but in a BioEMU simulation, again, let me tell you, BioEMU is free for academic use. You can use, anybody can use, but provided you have a hand in, a bit of coding and bit of Linux environment. I myself have run BioEMU. It takes less than 1 GPU hour. So 10 GPU years to 100,000 speed up, 100,000 times speed up, and less than 1 GPU hour. It is reproducing almost the similar free energy surface. As you can see, we can see minimas, we can see deep minimas, global minimas, how the protein folds. And finally, you can see the structure of these two proteins, same protein folded. using one MD simulation and using one biome simulation. So as you can see, the folded structure is not much different. It is almost same, maybe 1 or 2% different. So does that replace the traditional computing services? I don't think so. I don't think so, because BioMu is, after all, it's a model. Maybe a particular protein will behave in a different pattern than the known models, maybe. So we cannot replace the traditional approaches, but we take the contemporary modern approaches of AI and try to. Imagine if I want to target this protein. Suppose this is a protein which is causing some disease. So if I want to target this protein, it will take me 10 years and to know the structure of the protein. And versus now, I can know within 1 hour what is the structure of the protein. So isn't it amazing? And not only that, it will give me the structural variations. How many minimas a particular protein can exist in. So I would say it's a very powerful technique, provided you know how to use it, and provided how they improve the model in coming time. So success rates is large scale motions 80%, local unfolding we can see 75%, hidden binding pockets we can see more than 70% of the time. So almost just like a traditional molecular dynamics simulation, the AI-based simulation, AI-based, it will give us similar results. So success stories and case studies. I will tell the most, most proficient success story of ML technique called RF diffusion, by which we can design proteins against another proteins. So where this thing is coming, protein targeting another protein, it happens with somebody's mic is.
Thank you so much, sir.
Hello.
Am I audible, sir?
Oh, hi, you are audible. So it's a technique, RF diffusion, by which so diffusion is a AI/ML technique by which we can actually develop proteins, protein binders against another protein target. So it's an amazing thing, according to me. So this led to the Nobel Prize to Dr. David Baker, who developed these ML techniques for protein design. So such a huge impact. So we can see notable AI drug discovery achievements like in silico medicine from Existence here, and then Atom-Wise, then Halicin antibiotic discovery. These are the drugs recently discovered, and they are in phase 1, phase 2 clinical trials. None of it in phase 3 yet, but phase 1, phase 2 clinical trials. So again, we see AI's role in COVID-19 drug discovery, where vaccine was discovered in a very record time, which does not happen as such. So the time was like that. So people worked very hard, and then they use a lot of AI technologies, and they came up with, there's a field called vaccinoinformatics. If anyone is interested, they can know more about it. And then what are the advantages? Traditional approaches 10 to 15 years, and using AI 3 to 6 years. Cost reduction, you see 2.6 billion, and now it is not even 1 billion. So 70% decrease in rates, prices. And then success rates, it was 10% earlier, now it is 90%, 80%. And then chemical space, traditional approaches, limited libraries we had. Now we have billions of compounds due to generative AI. Then prediction, accuracy, and which is again to the experimental level. It is so nice. Previously, it used to take years per compound. Now it is days or weeks. Even in weeks, you can come up with novel compounds. Again, challenges. It also has challenges. It depends on data quality, how much data, how good data you are putting in. Similar kind of results you are going to get. So in computational, we use a jargon called crap in, crap out. Garbage in, garbage out. If you put bad data, you get bad. And then again, model interpretability is there. Not everyone can do a AI model interpretable. So AI models are still like black box. So transparency is not there. And then biological system complexity. Now they are working on simple problems, but we have to see how much it performs when more complex challenges come into picture. Future trends in AI and drug discovery with computing, quantum computing coming in, the things are going to really change in coming 30 to 40 years. There's going to be a huge paradigm shift when computing, quantum computing comes into picture. So it provides an exponential speed up for complex calculations. And then multimodal AI integrations, and then combining genomics, imaging, clinical data. So we are going to get the real picture very soon using AI. Personalized AI-driven medicine. Each person, they will have a different medicine, a different compound. All the compounds, all these drugs will be safe drugs. So individual genetic profiling can be done, and then precision drug design can be done, real-time treatment optimization, and then continuous learning, and democratization of drug discovery. This is the most important. Now everybody can do a drug discovery. Everybody, provided you know. So market growth, as you can see, the growth it is showing around 30% annual growth rate, cumulative average growth rate. So the growth rates are very high. Around 150 companies using AI for small molecule design are there in the internationally. I'm talking about now. Major pharma companies are making partnerships with the AI startups to design more efficiently, faster. So key takeaways, what are the key takeaways? Like, is, as we understand, it's fundamentally transforming drug discovery. And we can see significant improvements in speed, cost, success rates. Investment is a lot of, although it is coming, but still it remains critical because investors are thinking that this is like a balloon. It's going to burst soon. But as I understand, it's going to remain for a long time. It has completely changed the way we are thinking towards any problem. So finally, as you can see, the success rates have improved a lot. The cost 2.6 to 0.8, and where the drug discovery timeline was around 13 years. So it is getting projected because none of the molecules are in phase 3 now. They're all in phase 2. Around 4.5 years, we are thinking that we are going to discover new drug. So with that, I would like to finish my presentation. You can ask questions if you have any. If I know the answer, of course, I will give an answer. Yes.
Now it's your turn. We had loved to hear you. Do you have any questions about computational drug discovery in the AI area?
If immediate questions are not coming, you can write an email to me. I am working with GLA, so suman.sinha@gla.ac.in. You can send me email or you can, if you are around, you can just drop in the campus, and we have a beautiful campus. We can have a chat and we can discuss.
Okay, sir. Sir, can we wait for 1 minute? If anyone of the audience can ask. Okay. Thank you so much, sir.
My pleasure.
As we have seen, AI is poised to revolutionize the field of drug discovery with its ability to analyze vast amount of data, identify patterns, and make predictions. AI can help researchers develop more effective and targeted therapeutics. Thank you, Dr. Sinha, for sharing your expertise with us today. Thank you, sir, for your insightful presentation on the role of AI in revolutionizing drug discovery. You have covered the challenges of traditional drug discovery, the evolution of computational methods, and the impact of AI on the field. Your discussion on AI machine learning methodology in computer-aided drug design has been enlightening. Thank you so much, sir.
My pleasure, sir. Thank you so much for calling me. And
thank you, sir. Thank you so much
that's
for
nice.
coming. And closing of the day 2, I'm grateful to our speaker, Dr. Soman Sinha, organizers, Dr. Devendra Pathak, President of IPGA, and Dr. Himanshu Chopra, Vice President of IPGA and Director of Rajiv Academy for Pharmacy, and participants for making day 2 a success. Let's look forward to another exciting day tomorrow. Until then, good night and thank you for your participation. I would like to thank you, our co-convener, Dr. Yogesh Murthy, for addressing. This closing remark acknowledges the efforts of the speaker, organizers, and participants, while also setting the stage for the next stage proceedings. Thank you so much to all. Thank you again. Now you can end the session.
Okay, Pandey sir.